Hello students, welcome back to the biology classes. Dear students, in previous lecture we have studied about the mutations and variations. Dear students, in this lecture we are going to discuss about the pedigree analysis. So dear students, let's start our topic. Dear students, pedigree analysis is the graphical or diagrammatic representation of the family history of an individual. And dear students, in the pedigree analysis, we record inheritance of some genetic traits. Okay, in this pedigree analysis is mainly used in case of human beings and domesticated animals. Dear students, here are some symbols which are used in uh, human pedigree analysis. Dear students, you can see that for normal females we use circle, for normal males we use square, and so on. So please watch these symbols very carefully. Dear students, sometimes a question arises that why pedigree analysis is used to study human inheritance. Dear students, we use pedigree analysis for the study of human inheritance because the generation time of human beings is very long. Okay, it is near about 20 years, and so the research workers are not able to observe 3 to 4 generations in their lifetime of humans. So, and also the number of offspring produced in humans is very small. Okay, and their homozygous traits are very few in case of human beings, and in case of humans, at each marriage combine alleles from the two different families. So that's why. Um, we use pedigree analysis. Beside this, many human traits are governed by large number of genes. We also have large number of chromosomes. They are near about 46. Okay, and there are also an impact of environment on human on the expression of genes in humans. So that's why in humans we use pedigree analysis. We are not able to use the experiments like Mendel used to study the inheritance of P on the human beings. So that's why it is easy way to study the human inheritance. Okay. Dear students, there are many importance of the pedigree analysis. First, the pedigree analysis is very useful for the genetic counselors to advise couples about the possibility of having children with genetic defects such as hemophilia or colorblindness. If an individual have hemophilia or colorblindness, so by studying the inheritance in his family history or in the we can the research the counselors are easily tell that what is the probability that the offspring may affect from these disease, okay, these defects. And it also indicate that Mendel principle also applicable to the human genetics but up to a certain level. Okay, and beside this, uh, by the use of pedigree analysis, they can indicate the origin of traits in ancestors. They also able to indicate that the harm caused by the marriage between the close relatives. Dear students, pedigree analysis mainly use two tools. First is the principle of probability, and second is elimination of alternative. So the pedigree charts which we are going to construct are mainly depend upon these two tools. Dear students, when we construct a pedigree chart for autosomal dominant trait, we have observed that the autosomal dominant trait never skip the generations. You can see this with the help of this diagram. You have what you have seen in this diagram, that in this diagram there are three generations of a family are represented with the help of symbols. and they are given the numbers in Roman: first generation, second generation, and third generation. Okay, 1, 2, and 3. Here in the first generation, parents are present, okay, which has given name 1 and 2. So first is female, which is represented by a circle which is dark, okay, whereas the second individual is male, okay, which is represented by a square, and that cannot have, or that is not affected. So here the female is affected whereas the male is unaffected, or we can say cannot show the trait, okay. And if it is a dominant trait, okay, autosomal dominant, here we use alphabet A to represent the autosomes, or for the dominant we write capital A, or for the recessive we write small a. So it is the probability that if the female is affected, it may have, it either have one capital A, or we can say the one dominant gene. So we use the principle of probability here. We write capital A, small a. We don't write here capital A, because if we write capital A, the all individuals of the second generation are affected, okay. But when we see the second generation, 50% individuals are affected or 50% individuals are unaffected. So probability is that the parent is female which is affected has genotype capital A, small a, okay, whereas the male is unaffected, and so that's why it has small a, because if it has capital or a dominant gene, then it is also affected, because it, this diagram show the, or this pedigree chart show the inheritance of autosomal dominant trait. So you can see that in the first generation, both female have capital A, small a, while the male has capital small a genotype. So that's why female is affected or male is unaffected. And in second generation, their offspring are shown, okay. These parents produce four offspring. Among them, two are daughter, or we can say the two are the female and two are male, okay. In this you can see that in the second generation, the first offspring, their first offspring is female, which is represented by the circle, and is unaffected, which is represented by small a. Similarly, their second offspring is male, okay, affected, which is affected, okay, that's why it is dark, okay, the square is dark, and the genotype is capital A, small a, whereas the third is also a male, third offspring is also male, which is represented by the square, and their fourth offspring is female, which is represented by the dark circle, okay. This is the second generation, where in third generation you can see that their third offspring for second generation is married to a female, unaffected female, okay, and they produce offspring which are represented in the third generation, and they produce two female offspring and one male offspring, and all are unaffected. So that's why they all have genotype small a. But their fourth offspring of the second generation is represented by a circle which is dark, okay, and it has genotype capital A, small a, and it is married to a male which is unaffected. So that's why it has small a genotype, and when they married, they produce three offspring which are present in third generation, shown in third generation, the individual 4, 5, and 6, okay, and among them two males and one female. Two male are unaffected, so that's why they have genotype small a, whereas the female is affected and shown by the dark circle, which has genotype capital A and small a. So this is the inheritance of autosomal dominant trait. Okay, you can see that there are some individuals affected in each generation. First generation, female is affected. In second generation, one male and one female is affected. In third generation, one female is affected. You can see that in autosomal dominant trait, uh, the trait cannot skip any generation. Okay, and second is autosomal recessive trait. Dear students, autosomal recessive trait may skip the generation. Here, an example is given for the sickle cell anemia. Sickle cell anemia is a autosomal recessive disorder. So here, in this sickle cell anemia, the one gene is affected, and here the autosome are represented by HBA and HBS. HBA and HBS. Okay, here HBA represent normal gene, whereas the HBS show the defective gene. Okay, but sickle cell anemia occurs when both affected genes HBS are present together. So at that time they show its expression, and individual is sickle cell anemic. So here you can see that in the first generation the parents are carriers. Okay, in autosomal recessive traits, or maybe autosomal dominant traits, the parents may be affected, may be unaffected, or may be carrier. Okay, so here in autosomal recessive trait, the in first generation, both parents are carrier. They have one normal gene and one defective gene, or we can say that the anemic gene. So they cannot show any characteristics of sickle cell anemia, but they carry the defective gene to another generation. So parents are unaffected, but when we see their offspring, okay, in the second generation, what we have seen, they produce five offspring. Among them, three are male and two are female. And you will see that the first male is unaffected, second male is affected, third female is unaffected, fourth female is affected, whereas the fifth male is unaffected. So the first male of second generation which is unaffected has genotype HBA, and second it may be HBA, HBS. Okay, any of the, it is a probability. Okay, it depends upon the probability. But second individual which is affected, it has both HBS and HBS gene. HBS, so it has both affected gene. Okay, that's why it's so sickle cell anemia. Okay, and third is female which is unaffected, so it has one normal gene, and it may have second normal gene or affected gene. So it may be HBA, or it may be HBA or HBS. Okay, fourth is affected female, so it has both affected gene HBS HBS, which cause sickle cell anemia. Okay, and fifth is male which is unaffected, so it has one normal gene HBA, and second normal or may be affected, so it may be HBA HBA or HBA HBS. Okay, and in third generation what we have seen, the third offspring of second generation marries to a male which is normal, so it either has HBA HB or HBA HBS. It is depend upon probability. And you have seen that in the third generation they produce uh, three offspring. Among them, one male which is affected and two female which is unaffected. So the affected male has HBS HBS genotype, whereas the unaffected females may have HBA HBA or HBA HBS. Okay, one affected, one normal, or both normal genes. So this is about the autosomal recessive trait. So autosomal recessive trait may skip the generations. You can see in the diagram easily, or in this pedigree chart easily, that in the first generation individuals are unaffected. They are only carriers, so they are not showing any trait. So here they skip the generation. But they show the trait or the defect in the second and third generation. So we can see that autosomal recessive trait may skip the generation, whereas the autosomal dominant trait cannot skip the generations. Dear students, now we talk about the sex-linked traits. Dear students, as we all know that humans sex determination is XXXY type. So the sex-linked genes are present on X and Y chromosomes. So dear students, the X-linked dominant traits are mainly more common in females, whereas X-linked recessive traits are more common in females. And similarly, if Y-linked traits are generally passed from father to the son only, because the Y chromosome is only present in males. So dear students, I hope you understand. Thank you.